Welcome to Inert Chemistry. In today's video lecture, we will discuss about the transport of ammonia. In previous video, we had discussed about deamination. During the deamination, ammonia is released from glutamate. Now this ammonia has to be transported to the liver so that urea can be synthesized from it. Now we see that how this transport of ammonia takes. First of all, we will see the metabolic fate of ammonia. As you know, glutamate acts as a collecting center of amino group from various amino acids. Then during the deamination reaction, this glutamate is converted into alpha ketoglutarate and free ammonium ion is released. This ammonium ion or ammonia formed is having different fates. It may either convert it to glutamate, glutamine and urea. Glutamate. As you know that this reaction of deamination is freely reversible. Glutamate can either be converted into alpha ketoglutarate and free ammonium ion, and the ammonium ion and alpha ketoglutarate may recombine to form glutamate. So the first fate of ammonia is that it may either convert it back to glutamate or can be converted into glutamine and urea. The second fate is the synthesis of following compounds. Ammonia can also be used for the synthesis of non-essential amino acids, nitrogenous bases like purines and pyrimidines, amino sugars and asparagine. This ammonium ion is also having great importance in maintaining the acid-base balance of the body. First of all, we see that how ammonia is converted back into glutamate and then into glutamine. Alpha ketoglutarate and ammonium ion, both these recombine and form glutamate. This reaction requires glutamate dehydrogenase enzyme and can either use NADP or NAD. It occurs in the liver. And what is the significance of this reaction? By the formation of glutamate, the NH2 group of glutamate becomes available for the formation of non-essential amino acids by transamination. Now we see the formation of glutamine. The glutamate and ammonium ion, both these combine with each other and form glutamine. It occurs in liver, kidney and brain. This reaction requires glutamine synthetase as an enzyme and requires ATP. The glutamine synthetase enzyme is a mitochondrial enzyme and it is a unidirectional reaction. Glutamate to glutamine formation is a unidirectional reaction. It cannot be reversed. This glutamine formed from the combination of glutamate and ammonium ion is then sent towards the liver. In liver and intestinal cells, glutaminase enzyme act on glutamine and form glutamate and free ammoniamine. Then this ammonia is incorporated into urea via urea cycle. This glutamine acts as a storehouse of ammonia. It is the major transport and storage form of ammonia. Ammonia is also removed from brain as glutamine. The glutamine can freely diffuse from cells. As you know that ammonia is highly toxic, so most of the tissues readily convert ammonia into less toxic forms. For example, in most of the tissues, when ammonia is formed, then these tissues take action and combine the ammonia with glutamate to form glutamine with the help of an enzyme, glutamine synthase. Then this glutamine is transported towards the liver via blood, and as it reaches to the liver, it is converted into urea by the urea cycle. But most of the tissues use glutamine or form glutamine to transport the ammonia towards the liver. But muscle cells, they form alanine. How it is formed? It is formed by the help of transamination reactions. In the muscles, the amino group from amino acids is collected in the glutamate. Then the glutamate transport its amino group to pyruvate and forms alanine by the help of alanine transaminase. Actually, what happens? The amino acid gives its amino group or transfer its amino group to alpha ketoglutarate and form glutamate. So the amino group from amino acids is collected in glutamate. This glutamate then transfer its amino group to pyruvate and forms alanine. This alanine forms then transported towards the liver, and in liver this ammonia is again regenerated and incorporated into urea. The urea formed is then excreted from body through urine. In the next video, we see that how to write this topic as a long question in exam. Thanks for watching this video. We will soon come with another video with a new topic, that is urea cycle. Kindly subscribe the channel, like and share.